Hello and welcome everyone in today's session. Good evening. So I am Surasree. I'll be taking this live session with you again today for week 7, Immunology week 7 live session. Okay, so in today's session we will be discussing about how lymphocytes, which are naive lymphocytes, how are they activated upon interaction with any antigen, okay, and how are they activated in presence of any antigen. So before going into that, I just wanted to discuss briefly about so in this week's lectures, if you have gone through this week's week 7's lectures, you will find that professor was discussing about the maturation and the activation of T lymphocytes, right? So what is maturation and what is activation? Can anyone tell me the difference between maturation of T cells and activation of T cells?
Maturation is which occurs in the thymus, and activation is when they get, there's an antigen and then they get activated. Is that right?
Exactly. You're right that maturation actually defines the differentiation of the T lymphocytes when they become naive T lymphocytes from the primary thymocytes or from the immature thymocytes when they are matured to become naive T cells. Okay. However, activation of T cells is a different context altogether. Activation means when these naive T cells, when they encounter any antigen, and then from the naive T cells they become activated. All right. So before going into activation, I'll just briefly mention two terms here, which was there in the maturation stage of the T cells, and they were positive selection and negative selection. So anyone wants to highlight what was positive selection, what was negative selection of the thymocytes? Does anyone remember? Yes, thymic epithelial cells, they present the self MHCs, right? Now then what happens? What is positive selection and what is negative selection? Why are these two terms different and why are these two terms important in the maturation of the T lymphocytes? Okay, if you are not well acquainted with these two terms, let me just briefly discuss with you for once. That is, in the maturation phase, I told you that the thymocytes are becoming matured, and from the double negative stage they will finally reach the single positive stage. Double negative means what? That means they do not express either CD4 or CD8. So since both of them are absent, they are called double negative cells. All right, and from that they will finally mature into single positive cells. That is, either they will express a CD4 co-receptor or a CD8 co-receptor, right? So either CD4 positive or CD8 positive. So in this period, thymocytes have to undergo a very rigorous selection process, and this selection process is guided by the thymic epithelial cells in the thymus, right? So what do? These thymic epithelial cells, they present self peptides. Self peptide means something which is not foreign to us. The peptides which are derived from our own cells, from our own proteins, these peptides are presented by the thymic epithelial cells with the help of self-MHC molecules, right? So MHC molecules are also very different, and it can be different from one person to other. So with the help of the self-MHC molecules, the self-peptides are presented to the T cells which are not matured yet, to the immature T cells, right? And what happens in this phase, the T cells, they may either interact or they may either recognize that yes, this is the self-MHC, and if they recognize the self-MHC self-peptide complex, then they will undergo positive selection. That means they are able to recognize the self-MHC molecule. So that is a positive selection. So only the T cells which will recognize the self-MHC molecules will undergo positive selection. So here I am just writing it down once. Positive selection recognize self-MHC peptide complex. Whereas negative selection is an event where if the T cells, the T lymphocytes, if they interact with the self-MHC self-peptide complex very strongly, which means if they are activated or if they are stimulated by the self-MHC self-peptide complex, in that case they will not survive. Why? Because these T cells will be harmful for our body, because they can recognize the self-peptide and can be stimulated by the self-peptide, which is something which is not required in our immune system. Our T cells are programmed such that they can only be stimulated in presence of the foreign peptides, right? The foreign pathogens. So therefore, if the T cells are strongly being stimulated by interacting with the self-peptide, then it is a no for the T cells, and then they will not survive, right? So in this case, stimulated by self-peptide, and they will not survive. Okay, so which is why they are negatively selected. So these two rounds of selection actually goes on before the T cells are matured, and after these two rounds of selection, that means the positive selection and negative selection, then the T cells will become the naive T cells. All right. Now these naive T cells, this whole thing was happening inside the thymus, right? Now these naive T cells will have to exit thymus, and they will have to reach the secondary lymphoid tissues, where they can now recognize any foreign pathogen, right, or any foreign peptides, right? So therefore these T cells, I'm just telling once more, these T cells are the T cells which can recognize self-MHC, right, because they are positively selected, and they are also not stimulated by the self-peptides because they are negatively selected, right? So after these two rounds of selection, they will finally reach the secondary lymphoid tissues. And remember this thing that the T lymphocytes, they recirculate through these secondary lymphoid tissues, the peripheral lymphoid organs, because whenever they will actually get in contact or encounter any antigen, they will need to be activated, right? So for this activation process, they move towards the lymph nodes, right? Now the entry what is the brief overview of this activation of naive T cells? You can see here in this particular slide that is, T cells, now these naive T cells, they enter the lymph node via this high endothelial venules, right? Now they are recirculating, all right, they are recirculating through the peripheral lymphoid organs, and they enter via the arterial blood, they enter the high endothelial venules. Now this entry process, we will elaborate later in the next slide how this entry process takes place. Now once the naive T cells enter the lymph nodes, what happens there is that the T cells will either be activated or will not be activated. So how does this distinction happen? Because some populations, so suppose there is an antigen presenting cell inside the lymph node, and this antigen presenting cell is presenting a foreign peptide on its surface, right, through the help of MHC. Now either there will be some population of the T cells, of the naive T cells, which will be able to recognize this foreign pathogen, right, and then these cells will be activated. The rest of the cells which do not recognize this particular foreign pathogen, they will not be activated. So the T cells which are not activated, they exit from the lymph node. This is the exit, you can see here. Whereas the T cells which are activated by the antigen, when they recognize this foreign antigen, what do? They start to proliferate and they start to differentiate inside the lymph node, okay, and also they will not be able to exit anymore. So here you can see that the T cells are interacting with the dendritic cell, right, which is presenting this foreign pathogen, and they will not, these particular T cells which are activated, they will now not be able to exit the lymph node for a period of time until they are activated, until they are differentiated. And once the T cells are activated and they are differentiated, so they will also develop the subsets of the helper T cells, cytotoxic T cells and all that. So after they are activated, now they will and that they are differentiated, now they will be able to leave the lymph node. Okay, this is the final exit of the activated cells, activated T cells. Alright, so this is the overall process, how this activation process takes place inside the lymph nodes. Now I will go in detail about this entry, how these T cells, naive T cells, enter into the lymph node. So if you remember, in one of our sessions, we discussed about how the dendritic cells will be able to enter into the tissues, right? So whenever there is an infection in any tissue, what happens, the dendritic cells which circulate in the blood, they come out of these blood vessels, right, to reach to the site of action, to reach to the tissue of action where the infection has been taking place. So there also I had shown you something very similar to this process, which was extravasation. If you remember that, extravasation was the entry of the dendritic cells into the, crossing the blood vessels to enter the site of action. Here also the lymphocytes enter into the lymph node via these high endothelial venules, HEV, in a very similar process. So what happens, if there are three stages of this kind of entry, this process. First one is rolling. These stages are also very similar to what we learned in extravasation. Okay, so first step is rolling. So what happens, the lymphocytes, the naive T lymphocytes, they roll on the, so suppose this is the high endothelial venule on which the T lymphocytes roll, right? And how does this rolling action take place? this rolling action take place by the interaction of these particular molecules, L-selectin, glycam 1 and CD34. All right. So what happens, the first step is the binding of this L-selectin to the lymphocyte to the sulfated carbohydrates. So these sulfated carbohydrates are glycam 1 and CD34. Okay, these are sulfated carbohydrates which are present on the cell surface of the high endothelial venule cells. And this is the first step where this L-selectin binds to the glycam 1 and CD34, which allows this rolling interaction to take place. So now the lymphocytes will be able to roll on the endothelial venule, right? The next step is the activation step. And what happens in this activation step is that there is a chemokine. You can see its name here, CCL21. This is CCL21, and this chemokine receptor is called CCR7. So this CCL21 actually guides these lymphocytes into the lymph nodes. So what happens is that the chemokine receptor, it binds to this CCL21, this particular chemokine molecule, which are produced by the stromal cells of the lymph node, as well as they are displayed on these surface of high endothelial venule cells. Okay, so the CCL21 binds to the chemokine receptor CCR7, right? And because of this binding of the CCR7 to CCL21, it signals, you can see here, this kind of binding signals the LFA1, right? So LFA1 is what? LFA1 is an integrin. So this activation is basically the binding of CCL21 to CCR7 activates LFA1. So what happens, there is a conformational change which occurs in LFA1, because of which it can now bind to ICAM1. This molecule is ICAM1 and this is LFA1. I'm writing it down. LFA1 and this is ICAM1. So these are the cell adhesion molecules, right? So because of this binding of integrin to ICAM1, the LFA1 to ICAM1, there is a very strong interaction that takes place, and which allows the lymphocytes to finally migrate into the lymph node by this process, diapedesis, right? So this is how the lymphocytes will be able to enter into the lymph nodes, right? So, and this step is the adhesion step. So you can see there are 3 steps: rolling, activation and adhesion. All right, so this is how the naive lymphocytes will be able to enter into the lymph node. So now next step is what? Next step will be the activation process. Okay, so before going into this activation process, just let's take a look at what kind of cell adhesion molecules help in this kind of entry. So these integrins are very crucial molecules in the adhesion of the T lymphocytes, right? So integrins are heterodimeric proteins and they have 2 chains. You can see here, this is the integrin. You can see there is a larger chain which is an alpha chain, and there is a smaller chain which is a beta chain, right? So this is a heterodimeric protein consisting of 2 chains, alpha and beta. And the LFA1, this particular integrin, there are several types of integrins, and this particular integrin, which is LFA1, it is expressed on all leukocytes, and it binds to ICAMs. LFA binds to these ICAM molecules, which are the cell adhesion molecules, and they are very important for the migration of the cells and interaction of T cells with antigen presenting cells. Okay, so they are present on either the high endothelial value and also on the APCs, right? So this is the interaction of LFA1 with ICAM1. Next, here we can see that LPAM1. LPAM1 is nothing but lymphocyte pairs patch adhesion molecule. LPAM1 is lymphocyte pairs patch adhesion molecule. Molecule 1. Okay. So you can, from the name you can understand that they will be able to guide the lymphocytes to the pairs patch, right, or to the mucosal associated lymphoid tissues, right, or the mold, right. So on the mucosal endothelium there is this molecule present which is MADCAM1, okay. So which is which interacts with the, so this is an, what is this? This is an adenosine molecule, okay, which interacts with this LPAM1 and helps this population of naive T cells to enter into the mucosa associated lymphoid tissues or the mold regions, right. Next, also in the effector T cells, the activated effector T cells, there is one molecule present which is VLA4. Okay, VLA4 is also an integrin. These three molecules are all integrins. Okay, LFA1, LPAM1, VLA4. VLA4 interacts with VCAM1 on the activated endothelium. Okay, and it is important for recruiting these effector T cells to the sites of action or the sites of infection. Okay, so this happens in the site of infection. The interaction of VLA4 to VCAM1. So you can see on the naive T cells there is one kind of integrin adhesion molecule interaction that is taking place. On the effector T cells there is another types of integrin adhesion molecule interaction that is taking place, right. So these integrins are very important for these T lymphocytes, whether they are interacting with the APC, whether they are entering through the high endothelial venule into the lymph nodes, or whether they are entering into the site of action or the tissue of infection, right, whether in the infected tissue for this effector function. All right, and this is happening in the mold region, and this can happen in the lymph nodes. I hope that is clear. Any doubts so far?
Ma'am, can we go to the first slide?
First slide, yes.
Yeah, so here in the lower one it is saying T cells activated by antigen present by the dendritic cells start to proliferate and lose the ability to exit. And here it is saying activated T cell differentiate to effector cell and exit from the lymph node. I am getting confused out here.
Okay, so don't get confused. First of all, when this activation process is taking place, then these T cells will not be able to exit until and unless they are fully activated, and also they are differentiated, right? Because these activated T cells, as you know, that there are several subsets of the T helper cells, the cytotoxic T cells. The T helper cells also have different Th1 cells, Th2 cells, Th17 cells, right? So all these subsets, all this differentiation will only take place in the lymph node only, right? So once the T cells, they come in contact with the antigen on the surface of the dendritic cells, right, or on the surface of the antigen presenting cells, they start this process of activation. So I will go in detail of what is this activation process, right? And then several cytokines help these T cells to differentiate into different subsets, right? And finally, when this differentiation process is over, then only these T cells will be able to leave the lymph node, not before that. So before that, what happens, there is one molecule which is a sphingosine 1 phosphate. Okay, sphingosine 1 phosphate is a molecule which actually helps the T cells to exit from the lymph node. And the T cells which are not activated, suppose like these T cells which are exiting, right, they follow the sphingosine 1 phosphate gradient, that is outside the lymph nodes, the outside these high endothelial venules, there is a increased concentration of sphingosine 1 phosphate which is followed by these T cells, and these T cells follow this path, they leave the lymph node if they are not activated. But if they're activated, so there is one receptor on the T cells which is a sphingosine 1 phosphate receptor, okay, which is down regulated when this activation process is taking place. Down regulated means what? That means their expression is reduced on the surface of the T cells which are being activated and differentiated, so that these T cells cannot follow this gradient of sphingosine 1 phosphate and then cannot leave the lymph node. But when this differentiation process is over, then this sphingosine 1 phosphate receptor is again upregulated and they can now leave the lymph node after this these differentiated and activated T cells now can leave. Are you getting my point?
Yeah, right. Thank you.
Okay, all right. So this is how the lymphocytes are entering into the lymph node. 1 minute. Let me just check the message. Inactivated T cell wait in the paracortex of lymph node for antigen presenting cell. No, there is no such waiting that takes place. I told you in the beginning only that the principal thing which happens in the lymph nodes is the recirculation of the lymphocytes. Okay, the naive lymphocytes, they keep on circulating through the lymphoid tissues, so that once they come in contact with antigen, they can get activated. But if they are not encountering the antigens, they can be recirculated from the one lymphoid tissue to the other. Okay, but there is no such thing that they can wait in the cortex region or the paracortex region. That is not what happens. They remain in the lymph nodes for a certain period of time and then they leave. Then they are recirculated. Okay, is that clear to you? Okay, all right. So now we will be moving on to the activation process. So what happens during this activation? So there is this interaction with the APC and the T cell taking place, right? The APC is presenting this antigen. This red one is this antigen, right? And this is the T cell. Okay, so the T cell has expressed this TCR, right, and also has expressed this co-receptor which is a CD4. So this is a CD4 plus T cell, right? So it has expressed this TCR along with this CD4 co-receptor, and this APC is presenting this particular antigen with the help of MHC class 2, which is recognized by this TCR, right? This recognition has been taking place, and with the CD4 also, you can see that the CD4 is also binding to the MHC class 2 as well as the TCR, right? So this is the first step which occurs during this activation process. The first step is the activation of the interaction between the MHC class 2 and the TCR with the in the presence of this foreign peptide, right? So the first step, which is the activation, is nothing but the interaction of TCR with self-MHC foreign peptide complex. Just give me 1 minute. Okay. So this is the first step, the interaction of the TCR with the self-MHC foreign peptide complex. This gives the first signal for the activation of the naive T cells. The second signal comes from the co-stimulatory molecules. Okay, the signal for this survival comes from co-stimulation. And if you remember, what is co-stimulation? Co-stimulation is this interaction of B7 with CD28, right? There are several other co-stimulatory molecules as well, but B7, CD28 are the most common ones. Okay, so co-stimulation. So this is the interaction of B7, CD28. All right. And there is another signal which is required for this kind of activation, which is the cytokine signal. So the differentiation, now that the T cells are activated, they will be differentiated on the basis of the cytokines. Right? These cytokines, there are several cytokines which help the T cell to differentiate into several types of the T cell or the subsets of the T cell. All right. So these are the three signals which are required for any naive T cell to undergo activation and finally differentiation. Is that clear? Do you have any question? Okay. Now what will happen if you do not have any question? I'm just proceeding. That is okay. So what happens in here is that the T cells, these are suppose activated T cells. These activated T cells, they produce a cytokine which is IL-2. Okay, IL-2 is produced by these activated T cells, and what happens is that due to this activation, of T cells, they secrete IL-2 and they also express high affinity IL-2 receptors. So these are IL-2 receptors. Anyone told anything? So they also express these high affinity IL-2 receptors, and what happens, obviously this IL-2 can bind to this IL-2 receptor on the same T cells, right? So on the T cells, so suppose this is the IL-2 receptor, the IL-2 will also bind to the IL-2 receptor, and this kind of binding of IL-2 to the IL-2 receptor, it helps in the growth of the T cells and also in the differentiation of the T cells. So this is how the differentiation of the T cells take place, right? So this is the activation and the differentiation process of the T cells. Now in this process, once the cells are activated, once they are undergoing differentiation, so after the production of IL-2, right, the cells are now they are proliferating, and also they are being differentiated. There should not be uncontrolled activation and proliferation of these T cells also. Like there should be a limit, like where to stop, where these production of T cells will stop, or where the T cells now will be able to leave the lymph node, right? So for that there will be there is a checkpoint that checks the production on the proliferation of these kind of T cells, and that is provided by this inhibitory molecule which is called CTLA4. Okay, CTLA4 is related in sequence to CD28. Related in sequence to CD28. Okay, and the CTLA4 binds B7 molecules. So here you can see that CTLA4 binds to B7 molecules with a much higher avidity than CD28. Okay, so the interaction, the affinity of B7 to CTLA4 is much higher than that for CD28. And what happens because of that is that activation of these naive T cells, it induces this expression of CTLA4. So once the T cells are activated, well activated, they are well differentiated, now they express this CTLA4 molecule on their surface. Because of which what happens, now these activated T cells, they become less sensitive than the naive T cells. Okay, because this CTLA4 will now deliver inhibitory signal. Okay, so you can see this negative sign basically means inhibitory signal, right? So it will now deliver inhibitory signal to activated T cells that please stop, no more proliferation or no more differentiation is required. All right, so which will restrict the production of IL-2. So now the activated T cells, they will not further undergo IL-2 production, all right, but the naive T cells, yes, it is called negative feedback, you are kind of right, and these naive T cells, they will be able to interact with B7 with the help of CD28, and they will further undergo activation and differentiation, right? But the activated T cells, they will stop this IL-2 production, all right, and this will actually inhibit the T cells to further differentiate. All right, so here we learned about this activation which occurs by the interaction of TCR with MHC, also the interaction of the co-stimulatory molecules B7, CD28, and the cytokines on the cytokine receptors, and the inhibition takes place by the interaction of the co-stimulatory molecules B7 with the inhibitory molecule CTLA4. All right, okay, now coming to the last topic for today's discussion, that is, once the T cells are activated, so they will either become a CD4 T cell or a CD8 T cell, right? So this will be a helper T cell and this will be a cytotoxic T cell, right? Now the helper T cell also has various subsets which you can see here in this diagram. Th1 cells, Th2 cells, Th17 cells, T follicular helper cells, and T regulatory cells, right? These are the several subsets of the CD4 effector T cells, right? The activated T cells. And what is the point of this kind of having different types of these T cells? What is the requirement? That so that it can act differentially on different targets. Okay, Now the Th1 cells, these ones, right? The Th1 cells, they produce these cytokines which is called IF and gamma. Okay, so remember, these differentiation into the effector T cells is all guided by the expression of different cytokines. Okay, the Th1 cells, they express this cytokine IF and gamma, which activates the macrophages. So the target cell is macrophage, right? And what happens, this macrophage will be able to kill the, upon activation of the Th1 cells, by the Th1 cells, the macrophages will be able to kill the dead intracellular bacteria, right? So the Th1 cells, the effect of this Th1 cells is exhibited on the macrophages, which kills the dead intracellular bacteria. For example, this Leishmania donovani, right? Mycobacteria. These are the very common infections which occur in our society, and these microbes enter inside the macrophages, right? They infect inside the macrophages, and these macrophages, they are induced to kill these intracellular bacteria by the help of Th1 signaling. Okay, next, coming to the Th2 cells. Okay, the Th2 cells, they produce cytokines IL4, IL5, right? And they also produce IL13. Okay, so what happens, this IL4, it activates the mast cells and basophils. Okay, and IL5 activates the eosinophils. So here I'm writing actually IL4. Eosinophils are activated by the signaling of IL5. Okay, and what is the effect of these Th2 cells? That is, they help to get rid of these helminth parasites. Okay, so they give this anti-helminthic immunity by activating the eosinophils, mast cells and the basophils. Alright, now coming to the Th17 cells. The Th17 cells, they express these two types of cytokines, IL-22 and IL-17. IL-17 can activate the epithelial cells at the barrier site, which produce these antimicrobial proteins, okay, antimicrobial peptides that kill the bacteria. Okay, and here you can see that the stromal cells will secrete GCSF, chemokines. These are the several types of chemokines, right? And the target cell is neutrophils. So Th17 cells, they activate these neutrophils to kill the extracellular bacteria. So the macrophages kill the intracellular bacteria with the help of Th1 cells by being activated by Th1 cells, and the Th17 cells activate the neutrophils to kill the extracellular bacteria like this Klebsiella pneumoniae or the fungi, different fungal infections. Alright. Next, coming to T follicular helper cells. T follicular helper cells is a subset which is not activating the other types of immune cells but only the B cells. Okay, the T follicular helper cells, they produce IF and gamma to activate the B cells. Okay, and they actually induce the B cells to produce strongly opsonizing antibodies. Okay, so that they can strongly opsonize the pathogens, right? And the follicular helper cells, they actually induce these B cells to produce IgGs, right? Moreover, the T follicular helper cells also produce IL4, which drive the B cells to differentiate and produce immunoglobulin IgE. Okay, so IgE IgE can also be produced, which arms these mast cells and the basophil cells for granular release. So IgE is important in allergic reactions, right? So and the allergic reaction is produced by the release of these histamine granules, right? So that is also a part of this T follicular helper cells. And also the most important thing is they allow the isotype switching and affinity maturation of the B cells. Okay, they induce the T cells to undergo isotype switching and produce different they actually induce the B cells to undergo isotype switching to produce different several classes of antibodies. Okay, and the last one is the T regulatory cells. The T regulatory cells are different because they are an inhibitory type of cell. So rest all the cell types you can see, Th1, Th2, Th17, T follicular helper cells, they activate the immune response or they help to enhance the immune response, whereas the T regulatory cells, they inhibit the dendritic cells so that they cannot so that there is no case of autoimmunity. Prevents autoimmunity, right? So that the innate immune cells do not act against the self antigens, right? So the T regulatory cells play this role of inhibition. Okay, they are an inhibitory cell type. All right, so these are the CD4 effector T cells. Anyone having any doubt so far? T follicular helper cells do not release histamine. Okay, T follicular helper cells, they can actually induce the B cells to produce IgE, and this IgE can direct the mast cells to release the histamine granules. Okay, so T follicular helper cells do not release histamines, but histamine is only released by the mast cells. Is that okay? All right. Anyone having any other doubt? Okay, if you do not have any other doubts, let's go on to the question answer sessions of today. Please everyone type your answers in the chat box. Yes, all of your answers is correct. The answer is all of the above, right? So for clonal expansion and differentiation of naive T cells, all these three interactions, TCR interaction, B7-CD28 interaction, and also the cytokines binding to cytokine receptors, all of the above interactions are required. So this is the correct solution. All right, moving on to the next question. This question is about CTLA4. the question says if CTLA4 binds to the B7 molecules on the antigen presenting cell, what will be the fate of the T cell? Whether it will be activated or inhibited or co-stimulated or both activated and co-stimulated. So I think you are confused. Let's go back to the slide once. CTLA4 inhibits B7 or CTLA4 inhibits T cell response. Right? Yes, the correct answer is B. The T cell will be inhibited. Okay? Excuse me? Can you stop sharing? Okay, so the correct answer will be B, the T cell will be inhibited. Does anyone still have any doubt in this question? Okay. So this is the correct answer. Moving on to the next question.
Sorry, madam.
Yeah, no problem. So this question says, which of the following T cell subset is responsible for B cell maturation and isotype switching? Yes, your answers are correct. The T follicular helper cells. This is responsible for B cell maturation. Okay, next coming to this question. Now, which of these CD4 T cell subsets targets the eradication of Mycobacteria, Leishmania donovani? Yes, the answer is A, Th1 cells. Okay, this question is choosing the correct combination between the integrins and the adhesion molecules. Let's see if you remember. Choose carefully.
B.
Okay. P, B, D, C. Many people are saying many things. Okay, let's solve this. LFA1 Let's see. LFA1 interacts with ICAM1 on the naive T cells, right? Next, LPAM1, which is the pairs patch associated molecule, okay, pairs patch adhesion molecule, it interacts with the MADCAM1 on the malt tissue, right, mucosa associated lymphoid tissue. And the last one is the VLA4 interacts with VCAM1 on the activated T cells, right, to help them to enter into the infected tissue. So the correct answer is 1C, 2A and 3B, right? So which is B? This is the right solution. Is that all right? And one last question that I wanted to discuss for today, actually somebody asked me in a previous session, that is, there was a she was having a doubt in one of the assignment 5 questions, and I hope this was the particular question. This was there in assignment 5 of this course run, and also this solution is also out, so you can check. But if you now if you can answer this question, what do you think? What will be the answer? Just to give you the context ones, DRA, DRB, these are the genes that code for the alpha and beta chains of the MHC class 2. HLA class 2, HLA means MHC only, right? HLA is another name for MHC. Okay, so you are saying C is the correct answer. Can you explain, anyone, why is C the correct answer? How do you arrive at this number?
By multiplying 345 with 45, madam.
Yes, exactly. Correct answer. So these are the several exons, right? The several genes which are present for MHC class 2, and of course you can understand that any combination, any permutation of them will be able to produce the MHC class 2 molecule. So there can be, of course, so this is alpha chain and this is beta chain. So if you multiply them, 345 into 45, this will give you the total number of possible combinations, which is 15525. Hence the answer is C, right? So any other doubts for today's session? Also in the assignment questions, I am not supposed to tell you the assignment answers until and unless you have submitted or the submission date is over. But if you have like after the solutions are posted, there are also the detailed solutions where from you can check if you have any mistake or what is the reason of your mistake. But if you still have any other doubts from the assignments whose solutions are posted, you can ask us in the live sessions. We can explain if anyone of you having any doubts over there also. Okay, any other doubts for today's session, or I'll end today's session. Okay, thank you so much for joining today. Next class, next session, we again meet on Tuesday, the same time, 7 p.m. Till then, bye bye, and keep on learning immunology. Bye. Thank you, everyone.